Okay. So, good afternoon. Welcome to week 10 live interaction session. So, for today's class, we'll be discussing about cytokines majorly and the hypersensitive reactions. So, considering that this is week 10 and it was quite an information intensive week, I have decided to only cover some topics for today and some of the remaining topics that were covered as part of this week but will not be possible to be accommodated in today's class. I will be taking that up in the extra session that will be conducted next to next week. I don't have the exact date. Maybe I can check it once. Yeah, I think it's on 20th, Sunday. So we can have the discussion there along with some extra questions that I'll try to prepare as well in advance. So type 2 and type 3 hypersensitivity reactions that I'll be discussing in that class. So with this, I will start the discussion for this week. Now, since there are no students in the class as of now because of the odd timing, I presume, it will be a sort of a monologue where I'll try to explain concepts, and if there are any doubts, please go ahead and ask them in the discussion session, or you can, keep them with yourself until we have a live interaction, an opportunity for a live interaction sometime later on. So I'll switch off my video because my internet is a little trouble today. But okay, so like I said, as part of week 10, the theme for today's class is on cytokines. But before we get started, I will be discussing the previous week's assignment questions. So just to quickly brush up, the topic we had covered in the past week was about the complement system, the way it works, what are the three pathways, where do they converge, what are the intermediate steps, how does the cascade operate, um, what are the type of antigens which are able to elicit each type of like each pathway of the complement system, how does it cooperate or coordinate with the other immune effector responses within the body, and how it helps in assisting, or how it gives rise to the name that it complements the existing system. We had discussed all of that. The three pathways, we had discussed how each of them work, how the enzymes involved in the pathway work, and how it, from each step, the subsequent step is supported by the or aided by the previous step, and how the entire system is operational. Now, with this in just in the with this in mind, let's go over the questions for the past week. So we were first asked about the complement system. So what is the initial complement component that is bound by the complement fixing antibody? So we have C1s, C3b, C1q, or none of them. So if you remember that entire complex, I can maybe go back to it for ease of understanding. Um, yeah. So this was the complex that we had talked about that initially binds to the antibody. So we have C1q and C1R2S2, which all together forms that giant macromolecule, and the globular heads of C1q are able to make contacts with the Fc region of the antibody that has recognized its cognate antigen. So therefore, the correct answer to the first question is C1q. So that is what binds. That is the first complement component that is able to bind to the complement fixing antibodies. Second, we were asked about anaphylatoxins. So what are those? They are smaller fragments that result from the complement cleavage, which binds to receptors on mast cells and blood basophils and induce degranulation. All of these following are anaphylatoxins except C3A, C3B, C4A, and C5A. So I'll quickly again take you back to I think this slide. Okay, no, just a sec. We had this tabulated somewhere. Hmm. Maybe we can take this as the reference. So if you remember the overall convergence of all three pathways of the complement system, you would remember that the main amplification step or the main rate determining step in this case was the formation of the C3 was the development or the cleavage of C3 by the C3 convertase to generate C3A and C3B. From there, C3b was then further involved in the formation of the C5 convertase, and C3b is it is crucial to the formation of the C5 convertase, and it can also diffuse away and then bind to immune complexes, etc. And there it functions as an opsonin. So taking that into factor, it is not functional as an anaphylatoxin, and therefore the odd one out from the options that we had been given was C3b. So yeah, that was the small answer to this question. So it does not bind to receptors on mast cells, but instead it has its own set of functions that it performs, apart from forming the major component of the C3 convertase to the C5 convertase. The protein which catalyzes the proteolytic cleavage of the complement proteins is A, a convertase, B, a protease, C, a complement enzyme, or D, a complement cleavage. Now, the fourth option is obviously not correct. There's nothing called as a cleavage. The complement enzyme, yes, it is, if you think about it, this protein which is able to catalyze a certain reaction is definitely an enzyme. And since it's part of the complement system, you could consider it to be a complement enzyme as well. And also because it is carrying out proteolytic cleavage, it is technically also a protease as well. So options B and C might also seem correct to you. Now, if there was another option where all A, B, and C were, taken into consideration altogether, then maybe that would be our correct answer in that case. But since that's not what we see here, and because the question is specifically addressing that protein which is only able to function in the complement system and is able to catalyze proteolytic cleavage, taking all of these adjectives into consideration, the correct option in this case would be a convertase. And depending on which step it is, its nomenclature would be different. Like we just saw, the C3 convertase or the C5 convertase, and so on and so forth. So therefore, the correct option in this case would be the convertase, which is option A. Now, the next question was about several of the complement components are A, glycolipids, B, cell surface molecules of lymphocytes, C, enzyme precursors, D, hormones, or E, antibodies. So, like we just talked about it, the key factor that operates within the complement cascade are all of these proteases or enzymes or complement enzymes that subsequently work in each step. So as a result, the correct option in this case would be the precursor of enzyme or pre-enzymes, which are able to function in this manner, and therefore they are the key complement components. Option C in that case would be correct. The cofactor, which is not involved in factor 1 mediated cleavage of bound C4b in the classical pathway is A, C4b binding protein, B, complement receptor type 1, membrane cofactor protein, or factor H. So again, I'll go back to the figure for easy recalling. If we come to the classical pathway, I think it was should have been there. Okay, just a second. Okay, let me just put it directly then here because it's about figuring out the odd one out in this question. So if factor 1 or factor I mediated cleavage of bound C4b does not involve factor H in this case, so that's the correct option. It of course requires C4b binding protein, complement receptor 1, membrane cofactor proteins. Factor H is not required in this case, and therefore that's the odd one out and the correct answer to our question. The classical classical and alternative pathways meet the complement component in which of the following steps? So we have C4, C4b, factor D, C5, and C3. Now, like I just showed in the previous figure, that all three pathways, not just the classical and alternative, but also the MB-lectin pathway, all three of them converge at one key amplification step, and that is the production of C3, and then its conversion, or then its proteolytic cleavage by the C3 convertase to subsequently redirect it towards the downstream steps. So that is the correct answer in this case. So C3 would be the correct option. The last few questions from past week. First, we were asked about what does the membrane attack complex in the complement pathway consist of? A. Coalescence, B. C3b3b, BB, OH, or C5, B6, 7, 8, and 9. So, of course, this is like very easy question. And definitely the correct answer in this case is that the MAC, or the membrane attack complex, which we are talking about in this particular context, that involves that particular pore forming structure, or that cylindrical structure made up of each of these components, C5b, 6, 7, 8, and 9, polyC9 rather, that altogether forms that sort of cylindrical structure which embeds itself into the wall, or into the cell membrane, or the uh, external membrane of the pathogen which has come in, and as a result it forms pores, and then it eventually leads to the eradication of the pathogen, and therefore the immune effector response is successful in that case, and therefore that's the correct option, option D. Then the complement proteins of the lectin pathway bind to dash on the pathogen. So for the MB-lectin pathway, we had talked about all the different types of antigens for each of the different pathways, and what triggers or what stimulates or initiates that particular reaction. In context of the MB-lectin pathway, carbohydrates, or lectins basically, are the trigger or the antigens which are able to trigger the downstream MB-lectin mediated complement system activation. So that is our correct option in that case, option C. Um, of the following, I'm coming to this question first. Of the following, which one is the most important function of the complex formed by the complement components C5, B6, 7, 8, and 9? And that is very directly related to the question on the left, that it forms the membrane attack complex and it allows perforation or pore formation within the cell membrane, particularly in this case, bacteria is being given as a reference. So bacterial cell membrane perforation leading to leaching out of intracellular components and eventual cell death, lysis and eventual cell death. So option D is correct in that case. And finally, the last question, we were asked about innocent bystander lysis. Now, while I did not get into that question directly, if you have gone through the lectures, you would remember this being discussed by Professor. So innocent bystander lysis, as like honestly, in this case, you can even attempt this question if you don't completely remember the distinction between each of these phenomena. But innocent bystander lysis, as in a cell that is not supposed to be killed, gets inappropriately or incorrectly killed in place of the target cell, and that is because of the complement proteins that kills the normal cell that is very close or very nearby to the cell that originally was intended to be targeted, and its eventual clearance. So option B is the correct option in that case. Then coming, yeah, I think that wraps up the assignment questions for the previous week, and then we eventually move on to the. Just a second. Yeah, we move on to this week's discussion. Now, just a second. Sorry for the disturbance. So now let us get started with the topic for this week, which is about cytokines and how exactly they work. So by the very, we have discussed it several times before, if you remember what cytokines exactly are. And we had talked about them being the intracellular communicators or inter and intracellular communicators, and we have highlighted their overall roles in the different types of pathways that we have spoken about till now. The most relevant or the most frequently discussed, I would believe, is the fact that we are talking about isotype switching when we discuss the different types of antibodies and how you can switch from one isotype or one subclass of an antibody to another, and how that is so tightly regulated and so not even tightly, but so effortlessly regulated by the cytokine which is coming in and which is dictating that, depending on this X pathogen, this is the pathway that needs to be triggered, and as a result, this particular subtype or antibody isotype would be the most efficient at clearing out such a pathogen. And there's a beautiful tapestry that operates in ensuring that its regulation is very smooth. So with Okay. So for cytokines, we talk about, let's define it a little clearly now, even though we know the basic function. So the immune response involves lymphoid cells, inflammatory cells, and hematopoietic cells. The complex interactions among these cells are mediated by a group of proteins that are collectively designated as cytokines. What do? This denotes their role in cell-to-cell communication. Now, what are cytokines? We keep saying that they are molecules that help in, intracellular, intra and intercellular communication. But what they effectively are is that they are low molecular weight regulatory proteins or glycoproteins that are secreted by WBCs and various other cells in the body in response to certain stimuli. These proteins assist in regulating the development of immune effector cells, and some cytokines possess direct effector functions of their own. So basically, they are glycoproteins or low molecular weight regulatory proteins. We say low molecular weight because it dictates how small they are, so their diffusion, their um, transport throughout the body would be quite easy, and in fact is necessary. They are usually, I think most cytokines are under 30 kilodaltons as well. They're very tiny, and they're very effective at doing their job. So what they exactly do here is that this is again just a schematic representation. So you have an inducing stimulus, whatever that might be. For now, let's just take it as a stimulus X, which is acting upon a cell which is capable of producing cytokines. Now, because of the stimulus, there is expression of that cytokine gene, the gene which encodes for that particular cytokine, that is being synthesized and is being transported out of the, or is being secreted out of the system, and then that cytokine binds to its cognate receptor on the target cell. So cytokine receptor interaction is occurring, and as a result, there is activation of a certain gene which is able to induce a downstream biological effect. So this is how it normally works. So you have secretion of cytokine from cell X, and then on cell Y, it acts upon it, and then it mediates its biological effect. Now, it's not always necessary. I think I've discussed this also previously, that it's not always necessary that cytokine production and cytokine activity is on. Okay, somebody switched on the light here. Just yeah. Sorry. So yeah, it's not always necessary that cytokine synthesis and cytokine action will be on two separate cells. It does not necessarily have to be the case. So under such circumstances, you might have a condition where it is acting upon itself, whereby the cytokine would have an autocrine action. If it is acting on a different nearby cell, then definitely that is a paracrine action. We have discussed it. And endocrine action is a third type, which I believe we might not have discussed before. Either way, it's okay. But whenever we say endocrine, it definitely involves some transport through the blood vessels, through the vasculature. So in this case, that is exactly what happens, is that after the cytokine has been synthesized and released into circulation, it will migrate or will move through the blood vessels to its destination site, which might be a very distant location. And then at the distant location, it will execute its activity, and therefore the action of the cytokine is not limited in a spatial manner. It can technically, if you think about it, can circulate throughout the body and then find its target site and then act upon it. All you need is that the cognate receptor, receptor for that particular cytokine must be present. With this, we now get into a little more detail about how they work. I think the images are a little hazy. Um, but yeah, they should be easy to understand. But yeah, I think that it's slightly hazy. Okay. You can get the clear images from the book. Everything is from Kuvi. I've cited it below as well. But yeah, just for now, just bear with me and let's focus on the concept for now so that it's easier to understand. Starting off with how cytokines work. So here, what we saw in the previous slide is that the types of actions that can be executed. Now, after cytokine has executed its action, its effective conclusion or the way in which its action is being executed can also be subclassified into different types. And these are attributes of the way in which cytokines work. What exactly do I mean by it? You might already be familiar with it if you've already gone through the lectures. But yeah, basically what it does is that what this classification is doing is that it is trying to show you the effect of the way in which cytokines work. The first type of action, as you can see here, is the pleiotropic action of cytokines. So after cytokine has been synthesized, in this case, for reference, we are taking IL-4. One particular cytokine, IL-4, is able to execute very, very different effects depending upon the type of target cell it encounters. So obviously, if you think about it, the type of action in this case would either be a paracrine or it might be endocrine in nature, depending upon where the target cell is present. But as you can see, the type of action it is eventually executing is extremely different. For example, in case of B cells, we see that it leads to activation, proliferation, and differentiation. However, for B cells, for thymocytes and mast cells, it is only able to trigger proliferation, right? So what you understand here is that despite the fact that IL-4 and IL-4 receptor are the only participants in this in executing this interaction or executing this effect, you can see that how it manifests is very different depending upon the target cell in question. So that what it effectively is understood as is pleiotropy, which means that IL-4 has pleiotropic effects depending upon the type of target cell it encounters. That's the conclusion from here. Now, apart from that, several other phenomena are also understood. Like for example, if you see here, we talk about other types which are redundancy, synergy, and antagonism. How is that relevant? Redundancy, if you obviously know the simple English term, is that it is when two things are effectively executing the same effect, so they are considered to be redundant, which means that they are not performing any unique function and therefore they are redundant. Similar to that, if you look here, you have an activated T helper cell, which is then differentiating or is releasing certain cytokines which are taking the B cell as its target and are leading to its proliferation. However, you can see that all three of these cytokines, IL-2, 4, and 5, all of them are eventually leading to the same action, which is proliferation of the B cell. Therefore, in this case, they would be considered to be redundant in nature because any one of them can substitute for the action of the other, and they are therefore not performing any unique function. Next comes synergy. Synergy, by the definition of the word itself, simply means that if you have a factor x, and you have a factor y, the effect of individual factors versus the effect of the combinatorial action of both these factors is much higher. So if factor X was inducing an effect of both factor X and Y are inducing effects which are say can be quantified as 1, the combinatorial use of both X and Y together would be greater than 1. It does not necessarily have to be 2, which means that it does not necessarily have to be additive in nature, but it is definitely synergistic. It is more than what each of them could individually perform. So in this case, that would be an example of that would be IL-4 and IL-5, which are being secreted by activated T helper cells. Again, they are acting upon the B cell itself. And together, when both of them are able to bind to a particular B cell, then that is able to synergistically allow or induce the class switch to IgE isotype of antibodies. now you might have a question in mind that why are we taking both these examples for redundancy as well and for synergy as well? The thing is that when we are talking about redundancy, we take one factor at a time. We are just saying that either of them could substitute for their action. However, for a synergistic effect to be manifested, both of them have to be present. That is, IL-4 and IL-5 must be present. And both of them must be able to at least simultaneously or sequentially be able to bind to receptors present on the surface of the B cell. And then that will allow class switch to the necessary antibody isotype. And the last attribute of cytokine, which is antagonism, it's you definitely are familiar with this terminology if you have even studied some basic ecology in the past, which is a type where we understand where we learn about commensalism, amensalism, antagonism, etc. So antagonism, as the name simply indicates, it is something which is antagonizing or opposing the effect of another factor. So what you see here is that IL-4 is the you're taking up IL-4 as the example in this case, which is normally supposed to induce the class switch to Ig, like we talked about it. So IL-4 itself was capable of performing this class switch, but IL-5 together helps in a synergistic effect and better or a higher propensity of class switching to IgE under those conditions. However, when interferon gamma is present, it is antagonizing, opposing, or inhibiting this particular reaction, and therefore it is blocking this class switch, which was previously induced by IL-4. So that's how it works. And also, this example, unfortunately, is a little it's split. It's an error because I was cropping out the images in this case. But basically, another type of effect of the cytokines is that it can induce cascades. We are already very, very familiar with it. But just for reference, you can see there's an activated T helper cell, which is releasing interferon gamma that is that is acting upon a macrophage which releases IL-12, and then that acts upon the activated T helper cells, and then it leads to the synthesis of more and more cytokines. So it's a cascade, but you can also see it's somewhat of a positive feedback loop. So more and more effect upon the activated T helper cells is being increased, and each subsequent step you can see that more and more of the downstream cytokines would be synthesized. So yeah, these are the different attributes or the different ways in which cytokines can work. Now coming to a little more detail about the cytokine. So what you see on the left here is that you take into consideration one particular macrophage. This macrophage is presenting its antigen, and then that is getting recognized, and that is allowing in the activation of a resting T helper cell into an activated T helper cell. That is the basic interaction where we are starting off. Now the question is that what are the different types of activities which can be mediated or controlled by the action of cytokines? The first one you see here, let's start from the top, is that macrophages under definite conditions upon the induction by the appropriate stimulus can lead to inflammation. We will be coming into the inflammatory response very briefly, but if you remember, we had discussed it very early on during the course, where we had talked about the four attributes: edema, erythema. So, there's redness, there's heat generation, all the WBCs, especially neutrophils, rush to that site to mitigate whatever pathogen has come in and clear it off from the system. So that is the first interaction, or the first, not interaction, but the first effector mechanism you can see being executed by a macrophage, which has presented its antigen to a T helper cell. Apart from that, you see that the macrophage is capable of acting upon adaptive immunity. It is able to stimulate hematopoiesis, producing cytokines for the differentiation of T helper cells so that it forms an activated T helper cell, is directly able to act on the resting T helper cell. So altogether, what is it doing is that cytokines are able to promote activation, differentiation, proliferation, and under certain circumstances, even the death of T cells, depending upon what is the stimulus, what is the surrounding environmental condition. Can it sustain the survival of this particular cell, or is it more crucial to prioritize nutrients and then clear out that particular cell from the system to conserve more energy? All of that information is relayed or is carried or communicated by the use of cytokines. So what they're effectively doing is that they are promoting all these activities, not just for T cells. Here you're just being given T helper cells for for reference purposes. But like you saw, T helper cells themselves are able to produce a very large amount of cytokines as well. And apart from that, they also act upon B cells, which we saw. They act upon other macrophages, dendritic cells, NK cells, and other leukocytes, altogether mediating the entire uh, communication network within the immune components of any living organism. Now, if we look at the functions of these, so this was an overview of how cytokines work, how can their actions or their effector mechanisms be classified. Now we come to the exact target and effects in a little more detail. So, sorry. So you can see, if you look at this table, again, I have put in a lot of tables because I think that's the easiest way to, grasp a lot of information in a short period of time, and it's easier to comprehensively compare also different attributes. So most of today's presentation is in the form of tables, which have been beautifully explained in Kubi. You go into details there, you'll understand it. I'm sure you'll understand it very easily. If not, we'll of course discuss it if necessary. But yeah, so like I was saying, that cytokines have the functional role of cytokines have been broadly grouped into two types: some cytokines of innate immunity and some of adaptive immunity. Now, the question is, how are they acting? Let's say, first, let's take into consideration who is secreting that cytokine, and second, where is it eventually binding, and who is it acting upon, and what is the effect of its binding. So let's start off with IL-1, interleukin 1. It is secreted by monocytes, macrophages, endothelial cells, and epithelial cells. And what its effect is that it is able to cause inflammation. It acts upon the vasculature or the blood vessels, causing inflammation, can act upon the hypothalamus to trigger fever, to eliminate pathogens, can act upon the liver to induce some acute phase proteins to clear off the infection. Next comes tumor necrosis factor alpha, acts upon macrophages, causes very similar effect. You can see it also causes neutrophil activation in this case. IL-12 by macrophages, dendritic cells, acts upon NK cells to influence adaptive immunity to promote Th1 subset. We will be coming to this towards the fag end of today's presentation. Following that, we have IL-6 acting upon macrophages and dendritic macrophages and endothelial cells. What it also does is it influences adaptive immunity by controlling proliferation and antibody secretion. So yeah, you can go over this. I don't want to read everything, but yeah, quickly, if you just go over this, interferons, because I will not be elaborating on it in today's presentation for lack of time. We will be taking it up in my extra class if that is necessary. But yeah, interferons alpha and beta, which are respectively being secreted by macrophages and fibroblasts, are necessary for the induction of the antiviral state in most nucleated cells. And what it does downstream is that MHC class 1 expression goes up and allows activation of NK cells, which are necessary for clearance of the viral infection from the system. So these are participants within the innate immunity. For adaptive immunity, we have, of course, a different set of interleukins or cytokines which are operational in this case. So IL-2, 4, 5, 25, TGF beta, interferon gamma, each of which are secreted by different types of cells. And eventually what they do are that it can promote T cell proliferation for AICD. What AICD, I don't think we've discussed yet, but yeah, it's another process for cell death. So maybe it'll come up probably in the next or next to next week. So we can discuss it then. And apart from that, it is also executing T cell proliferation and other effector functions. So some conclusion from here is that each type of cytokine has a definite cell that is secreting it, which is the original cell or the mother cell, and then there's a downstream target cell on which the action is being executed. One confusion, in case you have, you will find that the terms cytokine and the term interleukin are often being very interchangeably used, and that is because they eventually indicate the same thing. Interleukin means something which is communicating between leukocytes, and therefore it kind of is similar to what the name cytokine also suggests, that it's a form of communication between cells. So you'll find interchangeable uses for both of these terminologies. Now coming to the molecular participants in the action of cytokines. So like we have repeatedly said that the cytokine binds to its receptor and then executes its downstream activity. So cytokine receptors here, you'll find that they have been mentioned to be majorly within majorly within these five families. In case there's any confusion, this is the more recent classification that I found in Kubi. But yeah, there are two ways in which cytokine receptor family classification has been done. One is based on their structure, and the second is based on their molecular mechanisms, which actually puts them into five categories. But it may happen that you might get certain books where the four classification system is still being maintained. It is okay either way. What matters is that you conceptually understand how each of these work and why each of them are relevant and retained within our immune system. So, talking about the five families, the first one is, of course, the immunoglobulin superfamily of receptors. If you remember, we have discussed the Ig fold or the immunoglobulin fold previously when we were talking about the antibody structure, where it involves these loops of 110 to 112 amino acids, and the ends of the loop are actually linked by a disulfide bond, and that is being consistently maintained in any protein which houses this particular fold is is said to have the Ig fold and can belong to the immunoglobulin superfamily of receptors. Now, for this family, you can see some example ligands being suggested here. We have c-KIT, a stemness marker. I don't know if you would remember even now, but very early on when, and maybe I can just go to the figure. Um, just give me a minute. When we talked about the development of cells, right, these immune cells from the bone marrow, yeah, it should be here. So, right. So if you see here, c-KIT is a surface marker which is present on the cell surface of any cell that actually has the stemness phenotype. And you can see that once its fate has been sort of decided within the thymus, its expression is gone. So that's actually how this classification has been made, or this statement has been made that c-KIT is a marker for stem cells, or a stemness marker is how we commonly consider it. So the c-KIT, so the c-KIT itself is a ligand that is actually responsive to the immunoglobulin superfamily of receptors. And then we also have IL-1 and MCSF. Now, NCSF, in case you're not familiar with it, is a colony stimulating factor, also a ligand, if you put it directly, and that is able to bind to the superfamily. The second type actually is the class 1 cytokine receptor, and commonly, as you can see, they are often also called as the hematopoietin receptor family. And it has a large set of ligands. And what you can see, there are two very key distinctive features. The first being that there are a number of conserved cysteines which are present within the receptor. And apart from that, there's one very distinguishing motif belonging to this particular class of receptor, which is WSXWS. And for this class, which is the class 1 cytokine receptor family, or the hematopoietin receptor family, we have a set of ligands. All these interleukins, I'm not reading them out because they're very large a number, but also you can see that apart from interleukins, it is also responsive to certain hormones, particularly the growth hormone and prolactin in this case, which is an unusual behavior, which you'll not usually find for most other receptors. But yeah, particularly for the class 1 family, this is a distinguishing attribute. Next, we have the class two family of cytokine receptors, and there you can see it's commonly known as the interferon receptor family, and there, of course, as the name suggests, the ligand in that case would be interferons like IFN alpha, beta, gamma, or IL-10, each of which are able to bind there. And while it looks very, very similar, obviously, as you understand, it is not, because it lacks this particular very distinguishing motif. But yes, it is also very rich in cysteines, and its conserved cysteines are not just limited to one block, or okay, maybe one domain. Yeah, you would consider this as a domain, but in both domains, you can see some conserved cysteines being present. And its set of ligands are definitely different from those of class 1 cytokine receptors. You would usually not find promiscuity in this case. By promiscuity, I mean that any ligand binding to any receptor, if that happens, then such a behavior is called as promiscuous. And if you remember, we had talked about this when we discussed MHCs and the way and the different properties of major histocompatibility complex molecules or MHC molecules. So in contrast to that, here you see very specific responses being attributed to very specific ligands because they are only able to bind to their cognate receptors. Coming to the next fourth class, which is the TNF receptor family, or the tumor necrosis factor receptor family. There as well, you can see some very distinguishing ligands like TNF alpha, beta, CD40, nerve growth factor, and the FAS ligand. Again, I am not sure if you're familiar with it, but FAS is a death-inducing ligand, a cell death inducing ligand, and therefore such a pathway. If you remember, we had talked about it in just the sorry. Yeah, in just this slide, that it can even trigger cell death as well. So that is being mediated and very strongly controlled by one of the examples being the FAST ligand and the FAST mediated death induction pathway. But of course, there are other parallel pathways which are operational as well. And finally, the fifth family is for the chemokine receptor family, and of course, its key distinction factor is its arrangement on the membrane. Like if you see here for the immunoglobulin and TNF families, you can see that there is one individual receptor molecule which is embedded on the cell membrane or on the cell surface. But in particular, the most distinguishing and the most fascinating fact about the chemokine receptor family is that it belongs to the GPCR class of receptors, which means the G protein coupled receptor family. And therefore, by virtue of its very name, it is a 7-pass transmembrane receptor, which means that it spans across the cell membrane 7 times because of this sort of um, I wouldn't say spiral, but yeah, this repetitive or this tandem way of folding. And at its end, towards its C-terminal end, you can see a blue colored G protein which is present. And that is the reason for that is because this does not have an intracellular or cytoplasmic tail. It's the information which is received by this particular receptor is conveyed via the G protein which comes downstream of it. Now coming to the ligands, you can see a different set of ligands here as well. IL-8, RANTES, MIP1, PF4, MCAF, and NAP2. So they are they're actually quite different and quite distinct from the other receptor families and their ligand interactions, but this is unique. Not unique, but it's a separate subclass. Let's put it that way. So these are all of the different receptor families. They are structurally quite diverse as well, but they belong to these 5 families of receptor proteins. I don't know if I got muted at some point. Now we will first focus on the class 1 cytokine receptor family in more detail. So the first class, if you remember, here like we saw it, this class 1 cytokine receptor family actually has this particular conserved motif and some conserved cysteines, and these are all the different ligands, and you can see that's actually quite a large spread. All of these different ligands are responsive to this family of receptors. Now the question is that when we talked initially about the different phenomena associated with cytokine response, we talked about these two terms, redundancy and antagonism. How is it actually operational? That is very well understood by these real life examples of cytokine receptor and their ligand family interactions. So the sharing of signal transducing subunits among receptors. What do you need to understand the keywords here? Signal transducing subunits of receptors. What does that mean? It means that this receptor is not a single molecule, but rather an assembly of multiple proteins and multiple components that are assembling together to exhibit whatever is its downstream activity. In this case, for the class 1 cytokine receptor, you can see that there are two chains. There's one alpha chain and one beta chain. The signal transducing component in this case, in most cases rather, is the beta chain, the much longer cytoplasmic tail containing what should I say, entity or component is definitely the beta subunit or the yeah, should be fine now. So I was talking about the signal transducing unit in this case. So let us take the example mentioned here, that is the GM-CSF receptor subfamily. Now I'll be going into more details about it later, but for understanding, if you just take the examples here, three different types of class 1 cytokine receptor families have been mentioned here. The first is the GM-CSF, which is granulocyte macrophage colony stimulating factor receptor subfamily. What does it have? It has a common beta subunit. Like you can see, there is a smaller alpha entity in for GM-CSF IL-3 or IL-5, but their beta subunits are quite long and they are the signal transducing components of this particular receptor system as well. The second type is for IL-6 receptor subfamily, and that has a different unit which is in common, which is called as GP113, which is a glycoprotein basically, glycoprotein 130, which is common in all of the different members of this particular subfamily of the IL-6 receptors. And finally, when we talk about the IL-2 receptor subfamily, you can see that there is a completely separate point of commonality there, which is a common gamma subunit. So you have alpha, beta, and in tandem, in conjunction, there is also a gamma subunit. And we can see that to be the common entity among all the different members of the IL-2 receptor subfamily. Now, don't worry, I'll be getting into more detail about the IL-2 receptor family as well pretty soon. But for now, you just need to understand the fact that there is a point of commonality, and usually it is the entity that has the ability to transduce the cell surface signal towards the interior of the cell so that its biological effect can be manifested. So let us now get into more specific examples. So consider, coming to the legend here, consider the GM-CSF receptor subfamily, which includes receptors for IL-3, 5, and GM-CSF as well. Each of these cytokines binds to a unique low affinity cytokine specific receptor consisting of a subunit only, consisting of an low affinity subunit which is permanently present. All three low affinity subunits can associate non-covalently with a signal transducing subunit, which is the beta chain in this case. The resulting dimeric receptor now not only exhibits an increased affinity for the cytokine, but it's also very efficient at transducing the signal across a membrane after it has bound its ligand or the cytokine in question. Interestingly, IL-3, 5, and GM-CSF exhibit considerable redundancy. IL-3 and GM-CSF both act upon hematopoietic stem cells and progenitor cells, and they activate monocytes, induce megakaryocyte differentiation. All three of these cytokines induce eosinophil proliferation and basophil degranulation with the release of histamine. So that is the downstream effect of the activation of this particular pathway. So the takeaway point here is that the signal transducing unit of each receptor subfamily uh, is able to associate with the other component of the receptor, and then they're able to form the functional receptor, which will communicate the necessary information within the cell. And that includes a number of different functions, activation of monocytes, megakaryocyte differentiation, etc. And also the induction of an inflammatory response. Yeah. Now here you can see the phenomena that I was just talking about. So you have interactions between cytokine-specific subunits and that signal transducing subunit of the cytokine receptor, which in that um, first case was beta. The schematic representation or diagram of the low affinity and high affinity receptors for IL-3, 5, and GM-CSF. You can for reference because this course might not exactly demand it, but eventually in the future, if you want to go towards research, particularly in immunology, then not just even immunology, for any research matter for that any research area for that matter, you need to be able to draw diagrams quickly, schematically, in a way that is easy to understand and that conveys the result in a crisp manner. So for such diagrams, you need to just learn how to demonstrate a phenomena like this. Like for example, you could demonstrate the fact that synergy or pleiotropy, etc. You can draw it out with that elaborate cell and all that diagram if you want, but quicker understanding comes from schemas like this. So what I was actually trying to highlight is that you represent three ligands in three different ways. So you have IL-3, 5, and GM-CSF. So what they are trying to say here, let's first understand that. So it is showing you low affinity and high affinity receptors. When each of these ligand specific receptors are present in isolation, we are calling them as low affinity. Yeah. However, when they associate with the signal transducing component, that is the beta subunit, then their affinity is considered to be high. Why is affinity important? That is because they will be able to bind to the ligand, will be able to retain it for a for long enough so that it can or is capable of inducing the necessary downstream effect. So for that, you need to have at least intermediate to good affinity for that downstream interaction to be or downstream reaction to be elicited. That is a high affinity receptor when it is cooperating or coordinating with the beta subunit, which is the signal transducing subunit of the receptor. Yeah. The second one is that is okay, yeah. The second one will show you an example of how you can see that uh, multiple interactions, like in this case, if we take IL-3, 5, and GM-CSF again, when they are forming this high affinity receptor, if the downstream effect, which is elicited from all three, is giving rise to the same biological mechanism, which is, say, proliferation in this case, then that is leading to an example of redundancy. So association of the cytokine specific subunits with a common signaling unit, beta subunit, allows the generation of cytokine specific signals despite the generation of the same signal by different cytokine receptors shown. The third subtype is the competition of ligand binding chains of different receptors for a common subunit. So that is an example of the antagonistic effect that I talked about, because on the cell surface, you will not have only one type of alpha or one type of beta and um abundant cytokines for them to serve as the uh, trigger for eliciting that particular pathway. But instead, what happens is that when this cytokine is present at a concentration which is limiting and which is crucial but limited, then under such circumstances, it might just happen that there is a preferential binding or there's a competition that occurs for binding to these ligands, and as a result, the um, binding of one particular factor. Let's go to the legend only, for example, the binding of IL-3 by alpha subunits of the IL-3 receptors is allowing them to outcompete the alpha chains of the GM-CSF receptor for beta subunits. So as a result, there can be, to some extent, there can be an antagonistic effect also uh, that can be observed here. Now coming to the more relevant example that already has been discussed, but I think is quite important because the IL-2 family of receptors is among the most extensively studied and a very interesting target in immunological research and has been for a while now, for like a decade or more. And the reason is because of its quite strange but very useful behavior. So let's go over the legend first. So a third signaling transducing subunit, which defines the IL-2 receptor family, includes receptors for IL-2, 4, 7, 9, and IL-15. The IL-2 and IL-15 receptors are heterotrimers. This is a new addition in comparison to what is already known. So they form heterotrimers. That means there is an alpha, beta, and a gamma subunit. So this consists of a cytokine specific chain and two chains which are beta and gamma that are responsible for transduction. The IL-2 receptor chain functions as the signal transducing subunit in other receptors, which are all dimers in this case. So what you see here, let's come to the schematic for better understanding. What you see here is that the alpha subunit, when it is present just by itself on the cell membrane, so then that receptor is considered to be IL-2R alpha. In such a or under such a condition, it has low affinity for the ligand and therefore it is serving as a low affinity receptor. And you can see that its affinity constant or Ka is 10 to the power 8 molar. Dissociation constant is also 10 to the power minus 8, which means that it dissociates quickly as well. And it is expressed by a number of cells. We'll come to that, majorly by CD4, CD8 T cells. Coming to the next subtype, which is the high affinity IL-2R that has all three entities and that has a very high affinity constant, but also has a significant dissociation constant as well. Third is the intermediate affinity one, where instead of alpha, you have beta gamma receptor that is able to bind to the ligand and that has intermediate affinity for the IL-2 ligand. And there you can see the subunits being composed of IL-2R beta and IL-2R gamma with a high affinity constant of about 10 to the power 7 and dissociation constant of 10 to the power minus 9. And the cells that actually express it are NK cells and resting T cells. So what we effectively understand from here is that the level of regulation of even cytokines is so subtle but so beautifully managed, is that while on one hand you will try to control the synthesis of the cytokine itself from the mother cell or from the original cell, how well that cytokine is able to execute its response is also being controlled by the target cell. So it can change or it can regulate the relative abundance of all of these different IL-2 receptor family subtypes, that is the low affinity, intermediate affinity, and high affinity form of the receptors, and then they can be studied in extensive detail. So it has been shown that actually the congenital disorder, which is the X-linked SCID disease, which is severe combined immunodeficiency syndrome. It results actually from a defect in the gamma chain gene that maps to the X chromosome. The immunodeficiencies that are observed in this disorder are due to the loss of all cytokine functions mediated by IL-2 receptors. So that basically sums up how the class 1 cytokine receptor family works. Now coming to what happens downstream of that. So till now we understood how the ligands work, how their binding can execute its effect. Not how it affects, but we just talked about the fact that the binding of the ligand to its cognate receptor has some defined downstream effects. Now the question is, what exactly happens there? How is the downstream? How are we reaching that downstream effect? So the key pathway that operates in this case is the JAK-STAT receptor or the JAK-STAT signaling pathway. So I'll simultaneously read out from the legend and also the figure for ease of understanding. So we start off, of course, with the receptor from the cell surface because that's where the stimulus must arrive. Just a second. So the cytokine receptor in this case, of course, is composed of separate subunits. There's an alpha chain required for cytokine binding and for signal transduction as well. And there's a beta chain, which is necessary for signaling, but with only a minor role in binding activities. Now, different inactive protein tyrosine kinases are associated with different subsets or subunits of the receptor. The alpha chain is associated with a novel family of protein tyrosine kinases, the JAK kinases or JAK family. Now, this is not very novel till date. Of course, it's an interesting area of research, but um, it has been around for a while. So, what we effectively understand here is that the effect of class 1 receptor binding to its ligand and whatever contributed there is leading to the eventual activation of the Janus kinase or the JAK-STAT signaling pathway. Now, JAK is for Janus kinase and STAT, of course, is for signal transducer and effectors of transcription. The association of the JAK and the receptor subunit occurs in a very spontaneous manner and it does not require the binding of the cytokine. However, in the absence of the cytokine, JAKs lack protein tyrosine kinase activity. Now, cytokines binding, which is what is happening here, this you see the default condition, and then you can see here that a cytokine has bound to the alpha beta receptor. Ligand binding also is supporting the dimerization of the receptor, which is allowing its autophosphorylation and then execution of the downstream signaling. So what it is doing here is that the cytokine induces the association of these two receptor subunits and there is activation. The ability of interferon gamma, which binds to a class 2 cytokine receptor to bring out the association of the ligand binding chain of its receptor has been direct demonstrated by X-ray crystallographic studies. So what happens downstream of it? When there is binding of the cytokine, what that is doing is that it is allowing the dimerization of the receptor and the not trans, but the autophosphorylation um, of some tyrosine residues which are present on the receptor and the enzyme as well, on Janus kinase as well. So in under such conditions, you would consider the JAK or the enzyme to be activated. Now, activated JAKs are creating docking sites for STAT proteins, which are transcription signal transducers and effectors of transcription. So, what they are doing is that these STAT transcription factors are able to dock onto this activated JAK. By docking, we mean they are able to lodge themselves on top of it. So what that is doing is that it is allowing their docking to occur by phosphorylation of some specific tyrosine residues on cytokine receptor subunits. Now, once these are activated, they phosphorylate specific tyrosines in the receptor subunits of the complex, and then members of this STAT family are able to bind to the phosphorylated tyrosine residues. Now, these stats are, let me just read this out. Yeah. The binding of stats to their receptor subunits is mediated by joining of the SH2 domain on the stat with the docking site, which is created by JAK-mediated phosphorylation of tyrosines on the receptor subunit. So here you have the receptor. On top of the receptor, there is phosphorylation, and this phosphorylated receptor tyrosine molecule is allowing the docking of the STAT factors. Now, after undergoing JAK-mediated phosphorylation, STAT transcription factors are able to translocate or they are able to move from the receptor docking sites at the membrane to the nucleus, where they will initiate the transcription of some very specific genes. While docked to these receptor subunits, STATs are able to undergo the JAK catalyzed phosphorylation of a key tyrosine. This is followed by the dissociation of the STATs from the receptor subunits and their dimerization. And then these STAT dimers are able to move to the nucleus, induce expression, and then they're able to do their job. So from here onwards, it's a very it's well known, and it is something that is true for any transcription factor for that matter. So basically, the distinction point is this area where initially JAK gets phosphorylated, allowing the docking of STATs, and then this phosphorylated or activated JAK kinase is able to dimerize form the transcription factor, which can localize itself into the nucleus, and then it is able to bind to its response elements and execute the activity. So yeah, that was the way in which signal transduction is carried out. Now, how are cytokines working in terms of cell-mediated immunity is, if you remember, I had said that we will discuss this. So majorly, its action, one of the major ways in which its action has been studied and is of great interest is the way in which a naive CD4 T cell is getting differentiated or is getting modified based on what it is encountering. So there are two subsets of helper T cells, which is, of course, as the name you can see, there's Th1 and naturally there's Th2. So the Th1 subset is responsible for many cell-mediated functions. This includes, just need a water, a minute. The Th1 subset is responsible for many cell-mediated functions, including DTH, which I'll be discussing briefly today, or delayed type hypersensitivity and activation of T cytotoxic cells. And it is also very important for production of opsonization promoting IgG antibodies. What do? They bind to the high affinity Fc receptors of phagocytes, and they're able to coordinate with the complement system, thereby merging the two together and giving, justifying the name of the complement system as well. And also, this subset is associated with promotion of excessive inflammation or basically hypersensitive response, and for tissue injury as well. On the other hand, when we talk about the Th2 subset, that stimulates eosinophil activation and differentiation, providing help to B cells and promotes the production of relatively large amounts of IgM, IgE, and non-complement activating IgG isotypes. It also supports allergic reactions. So that's a different version of the hypersensitive system. So you can see that basically the T helper cells have a major role to play in inflammation, allergy, and hypersensitivity associated responses. Just to schematically see how it exactly works. You can see again the same interaction between a macrophage and a naive T helper cell. We had seen this a couple of slides back as well. And you can see all of the different networks that are operational in this case. First and foremost, you can see that the naive CD4 T cell after this interaction is able to release IL-2, which can act directly upon itself for more and more activation um, of CD4 plus T cells with this particular uh, genomic rearrangement. Added to that, you can see that this particular macrophage and then releases certain cytokines like IL-12, which act upon the Th1 subsite that has these defined functions, including but not limited to delayed type hypersensitivity, activation of macrophages, and then certain opsonic or complement fixing IgGs might also be involved. On the other hand, when there is enough of this, what can happen is that a positive feedback loop which operates can often come into play, and that positive feedback loop is often also found to be concordant with the suppression of the other subtype. So what are we understanding here is that the IFN delta, IL-2 and TNF beta, which are being released by the Th1 subset, will promote more and more of its own synthesis, while at the same time simultaneously it can inhibit the production or differentiation into the Th2 subtype. So that's how they actually these cells or the immune system at large is able to minimize energy expense and rule out redundant activities if possible. So yeah. So on the other hand, we see that for the Th2 subtype, there is IL-4 action, which is able to on the Th2 cells, and then that is able to release IL-4, 5, IL-10, and IL-13. That is allowing eosinophil activation and production of IgE and some other antibody classes. Again, there's a positive feedback which operates there as well. Now, cytokines are, we understood a little bit about the mechanism, the molecular pathways which are involved there, the um, the specificity which is maintained with respect to the ligand as well. Now, finally, how have they been actually used? All these studies that we are talking about, how have they been practically interpreted? So that is in terms of its clinical usage. So you can see a number of drugs which are actually operational in this domain. You have Enbrel, which is a chimeric TNF receptor IgG constant region containing agent used for rheumatoid arthritis. We have Remicade. You might have heard this name. Which is a monoclonal antibody against TNF alpha receptor to cut down on the amount of inflammation which is being used, which is being generated or which is in place in such patients. And its clinical application is with respect, of course, rheumatoid arthritis, like I just mentioned. Then you have interferon alpha 2A or alpha 2B used as antiviral cytokines, clinically used for hepatitis B, Kaposi sarcoma, hairy cell leukemia, or hepatitis C, melanoma, etc. So antiviral cytokines have been looked into and in a large number of cases also approved for clinical use. Then you have interferon beta, which is used for multiple sclerosis. That is almost always considered to be a terminal disease because it's very difficult to completely recover from it, and therefore its nature is majorly sort of palliative, if you want to say so. Then comes Actimune, which is for interferon gamma, used for chronic granulomatous disease, osteopetrosis. You can look, I think I had mentioned chronic granulomatous disease. If not, you can look into it. You'll find a lot of information online. And also osteopetrosis. Then comes Neupogen, which is for GCSF, which is hematopoietic cytokine that stimulates the production of neutrophils, reduces infection in cancer patients and cancer patients that have been treated with chemotherapy. Leukin for GM-CSF again stimulates production of myeloid cells. After a bone marrow transplantation has been carried out, it actually aids such patients. Numega stimulates production of platelets, and Epogen for erythropoietin stimulates RBC production. So, oh, sorry. So, yeah, I think that completes it. Yeah, these are the clinically used cytokines which have been approved and in a large number of cases also implemented for therapeutic usage. Now, coming to the second topic, I will not be getting into detail about this one. Let me second, let me check it. Yeah, we don't have a lot of information. We have like 6 slides on it, so we'll wrap it up quickly. Um, just a second. Yeah. So hypersensitivity, that's the topic. What does it mean? So an immune response, I'm reading out the legend, an immune response mobilizes a battery of effector molecules that act to remove antigens by various mechanisms. Generally, these molecules induce a localized inflammatory response that eliminates the antigen without extensively damaging the host tissue, which is what is expected from the immune system and what would be the ideal scenario. However, under certain circumstances, this inflammatory response can have deleterious effects, resulting in significant tissue damage or even death. This inappropriate immune response is termed as hypersensitivity or, in a large number of cases, allergy. We will preferably use the word hypersensitivity because that is semantically correct and it's just more acceptable, let's put it that. Now, although the word hypersensitivity implies an increased response, the response is not actually heightened all the time, but instead it might be an inappropriate immune response to a particular antigen. As it became clear that several different immune mechanisms give rise to hypersensitive reactions, Gell and Coombs, who are the pioneers in this particular work, so Gell and Coombs proposed a classification scheme um, in which hypersensitive reactions are being divided into 4 types. The 3 types of hypersensitivity occur within the humoral branch and they are mediated by antibody or antigen-antibody complexes. First is type 1, which is IgE mediated, antibody mediated, type 2, and immune complex mediated, type 3. A fourth type of hypersensitivity depends upon reactions within the cell mediated branch and is termed as DTH or delayed type hypersensitivity. Each one has very distinct mechanisms, cells, and mediator molecules, and that's what we are addressing here in this schematic representation. First one, of course, is IgE-mediated hypersensitivity. What happens there? You have an antigen which is inducing the cross-linking of IgE bound to mast cells and basophils, and that is being accompanied by a release of vasoactive mediators. So that's what you see here as well, the allergen binding and allowing cross-linking of antibodies, or particularly IgE antibodies present on the surface of mast cells and basophils. What that is doing is it's leading to degranulation and release of vasoactive mediators. Vasoactive mediators means those entities or those molecules which are able to execute some action upon the vasculature of the individual. Usually, the typical manifestations include A. Systemic anaphylaxis, B. Localized anaphylaxis, such as hay fever, asthma, hives, food allergies, and eczema. These are the common manifestations, as you can see. Second is for type 2, which is IgG mediated cytotoxic hypersensitivity. So, antibodies directed against cell surface antigens mediate cell destruction via complement activation or ADCC. So, ADCC and the way in which it works, I will not be discussing today, again, because it will take a lot of time and a separate topic for, separate zone for discussion as well. But yeah, I will be taking it up pretty soon. We have like 2 weeks left, so we can discuss it then. Don't worry about it. But basically, if you just go over the overall idea, what you're seeing here is that you have antibodies which are directed against certain cell surface antigens. What it is doing is that it is destroying those cells. So this is complement activation. This is ADCC. So you have this particular cytotoxic cell with its Fc receptors, and this is the cell target cell surface antigen, which is getting recognized by the cytotoxic cells receptor, which is an antibody in this case. And that antibody dependent cell cytotoxicity means that the cytotoxic cell will now kill it directly. Alternatively, you have the same target cell with the same surface antigen, and to it, the antigen specific or cognate antibody will bind. And to its Fc region, we have the binding of C1q and activation of the complement system, which will lead to removal of that from removal of that particular mm, target cell. Typically, where is the hypersensitivity associated with this observed? Blood transfusion, erythroblastosis fetalis, and autoimmune hemolytic anemia. So I will be discussing erythroblastosis fetalis and the basic blood type matching or blood transfusion associated hypersensitivity very soon. Not in today's class, but very soon. The third type is for immune complex mediated hypersensitivity. So, as the name suggests, of course, there has to be antigen antibody interaction. These complexes are deposited in various tissues and they are able to induce the activation of the complement system and then ensuing inflammatory response mediated by massive infiltration of neutrophils. How is it observed? Usually, it includes localized Arthus reaction, generalized reactions like serum sickness, necrotizing vasculitis, glomerulonephritis, rheumatoid arthritis, and systemic lupus. So these are different physical manifestation or disease manifestations of this hypersensitivity, type 3 hypersensitivity response. Fourth is for type 4, that is cell-mediated hypersensitivity. So you have a sensitized Th1 cell, which is releasing certain cytokines. So again, this is your antigen binding to, which is being presented and which is sensitizing a Th1 cell that is releasing certain cytokines, which are activating some macrophages or TC cells, mediating cellular damage. Typically, this manifestation is seen for contact dermatitis, the most common example cited in this case. But apart from that, we also have tubercular lesions and also for graft rejection. So for transplantation, this is the most commonly studied, rejection response for, for any organ transplant. So we are just going to elucidate the type 1 hypersensitivity response a little bit more. So First, you can see that there is exposure to a particular allergen shown here in green, which is activating the B cell, which has the antibody specific to it. Again, just to reiterate, type 1, 2, and 3 are antigen antibody interaction dependent. And therefore, there is activation, there is internalization um, of that particular allergen and its presentation to via MHCs, and then that is getting recognized by the T helper cell, and then that along with IL-4 is also being assisted, and then that is allowing its differentiation into memory cells and plasma cells, and specifically plasma cells that are secreting certain allergens, and these allergens are specific. These allergens have sensitized the B cell in such a way that it will undergo class switching to specifically produce IgE, which are the most sensitive and the most efficient at eliminating an allergen and eliciting an allergy specific response. So these IgE, which are specific to that allergen, will then go and bind to a sensitized mast cell, and that mast cell will undergo degranulation when the allergen binds to it. So what this is doing is that the IgE from this, which is being released by the plasma cell, is binding to the mast cell. This mast cell is only sensitized till now. And as a result, what will happen now is that when this mast cell, the sensitized mast cell, is encountering the allergen, then it is undergoing the necessary response. So what this is doing is it's introducing an additional step, which will validate the accuracy of the response as well. And a non-specific response is not being unnecessarily stimulated at all points of time. So when there is again a secondary exposure to that allergen, then there is degranulation of the mast cells and the release of these vasoactive amines. What exactly they comprise of, you might already be familiar with. But yeah, what it does is it acts upon all of these different cell types, and then it is an overall systemic issue that starts as a response to a particular allergen which has invaded into the body of the individual. There are some key mediators that are involved in executing this type 1 hypersensitive response. So, of course, you have histamine and heparin, which increase vascular permeability and carry out smooth muscle contraction. Serotonin as well does the same job, and these are considered to be as the primary mediators of type 1 hypersensitivity. Apart from that, we have eosinophil chemotactic factor for eosinophil chemotaxis, and same for neutrophils as well, and a large number of proteases also, which are responsible for bronchial mucus secretion, blood vessel degradation, and generation of complement split products. So these are the mediators of type 1 hypersensitivity, or the primary mediators of type 1 hypersensitivity. When we talk about the secondary factors or secondary mediators, we have platelet activating factor, we have leukotrienes, which are increasing the vascular permeability or contraction of the primary smooth muscles. Then you have prostaglandins, bradykinin, and cytokinins. Each of them have their own uh, mode of action. So it just you can go through it for your own reference. You don't necessarily have to remember everything there. Now, this shows the test, which is usually done to detect if a particular person is allergic to a certain pathogen. So you can see that there are, little, um, how do I say? It's like a little injection or a little introduction of a particular pathogen, and they are being made at on the arm or something so that the response is quick and accurate. So control, there is dust, mite, grass, shrub, etc. So there are intradermal injections which are being done into the forearm. It's also often done in the back because that gives you a bigger surface area to test for a large number of pathogens. And as a response, what is observed is a wheel and flare response. You can see some of it for grass as well. And you can see that it has developed where grass was injected, and such an individual would be considered to be allergic to grass. And some drugs that are actually used to treat type 1 hypersensitivity. The most common one of these, which most of you would be, I assume, familiar with, is the group of antihistamines, which work against histamines. They block the H1H2 receptors on target cells. Cromolyn sodium, which blocks calcium influx into the mast cells for their degranulation. Theophylline prolongs high cyclic AMP levels in mast cells by inhibiting the phosphodiesterase, which cleaves cyclic AMP to 5' AMP. Epinephrine for adrenal or adrenaline, which stimulates cyclic AMP production, and cortisone, which reduces histamine levels. So yeah, these are the these are some of the drugs used to treat type 1 hypersensitivity. And very quickly, I'll just end with type 4 hypersensitivity. Just a second. Yeah. We'll be going into type 2 and type 3 later on, like I mentioned previously. But for type 4 or the delayed type hypersensitivity response, intracellular pathogens and contact antigens are considered to induce this. When we talk about intracellular bacteria, we have Mycobacterium tuberculosis, Mycobacterium leprae, Listeria monocytogenes, Brucella abortus. Intracellular viruses like smallpox virus, variola, herpes simplex virus, or measles virus. Intracellular fungi, Pneumocystis carinii, Candida albicans, Histoplasma capsulatum, Cryptococcus neoformans, intracellular parasites like Leishmania, contact antigens like picryl fluoride, hair dyes, nickel salts, poison ivy, and poison oak. We'll be looking into poison oak pretty soon. But yeah, these are the types of stimuli. And what exactly happens in that response is that it occurs in two steps. The first, as you can see, is a sensitization step, and the second is an effector phase. In the sensitization phase, you have an antigen presenting cell carrying that intracellular bacteria, which it is presenting on its surface, that is interacting with the CD4 positive T helper cell that is releasing certain cytokines, and then it is differentiating into Th1 cells generally. Now, DTH or delayed type hypersensitivity mediating cells include Th1 cells generally and very, very occasionally CD8 T cells as well. In the second effector phase, you can see that after there is subsequent exposure of these sensitized Th1 cells to the antigen, the Th1 cells will secrete a variety of cytokines and chemokines, which attract and activate macrophages and other non-specific inflammatory cells. Activated macrophages will become more efficient at presenting their antigen, and therefore they would help the DTH response and would be the primary mediators of this delayed type hypersensitivity reactions. And in the last slide, we are seeing here the way in which poison oak is able to induce an allergy in this case. So this is the development of delayed type hypersensitivity reaction after there's a secondary exposure to poison oak. So cytokines like interferon gamma, macrophage chemotactic factor or MCF, and inhibition factor MIF released from Th1 cells actually mediate this particular reaction. And tissue damage results from the lytic enzymes that the activated macrophages have released. So this is your allergen in this case, poison oak or toxicodendron radicans, which releases pentadeca-catechol that enters into the skin, taken up by APCs like Langerhans cells, which are then able to present that particular pathogen, and that is sensitizing the Th1. Such a sensitized Th1 is releasing all these three ligands, MIF, MCF, and interferon gamma. And then the tissue macrophage is also assisting that, and then that is going back into circulation. The tissue macrophages are actually able to counter that, and there is a hypersensitivity response against it. So this there would be tissue damage because of the lytic enzymes that these are releasing, and overall there would be an allergic response being elicited within the system. So with this, yeah, I come to an end for my presentation today. So let me just try switching on the camera. Little dark here. Let me just, yeah. Okay. So I have kept it a little brief because only one other person joined and then they left pretty soon. But to sum it up, basically today, what we have discussed, first we looked into the questions for the previous week and for the previous week's assignment. And then we went through the basic ideas that were discussed as part of this week. We went to the hypersensitivity response a little later. But basically, we had started off with the way in which cytokines work, the different ways in which they execute their action, the types of reactions that that combinatorial action of cytokines can execute, some distinction between the different cytokine receptor families, how they work, what are their ligands, what triggers them, and yeah, and I think I had some questions here as well. Okay, not today. Maybe I can include them in the final session. But finally, we discussed hypersensitivity and the different subtypes of hypersensitivity, as was elaborated by Jill and Coombs. And yeah, I think that was pretty much it. We will get into more details about type 2 and type 3 again. But that was the basic idea for week 10. We will have three more live interaction sessions for week 11 and week 12 as per usual. For week 11, the session is on Saturday from 3 to 5 p.m., 5th of October. And for the following week, I'll be updating it pretty soon. And finally, the extra session will be for a maximum of 2 hours. So if anybody joins, it's very good. If not, I'll upload the video lecture anyway, and you can refer to it for your own understanding. So thank you so much. I'll be ending the class for today. Have a good day.